I want to thank you, Stephen, and organizers for inviting me to give a little talk in this series, and it's actually very, very interesting, and I'm really enjoying it. I just have to say at the outset that I may be the only person in this audience who has not contributed a structure to the PDB, because as you'll see, throughout my career, I've always been more sort of engaged with intrinsic disorder rather than order. So we'll talk a little bit about that today. And what I'm going to talk about is investigating alpha-synuclein amyloid seeding processes by NMR. No, sorry. I can't seem to. Okay. So as Andy told you, so firstly, our lab has been working for many years on trying to understand the fundamental mechanisms of amyloid formation and aggregation in neurodegenerative disease. And a big focus in our laboratory has been to study alpha-synuclein, which is the primary protein component that's found in patients with Parkinson's disease. And of course, you all know that these diseases are really quite awful and very prevalent, and very, very unfortunately, there really still are no effective therapeutic strategies to combat Parkinson's, Alzheimer's, et cetera, et cetera. So it is really still an area of tremendous concern, as well as cost. So what I've drawn here are two cartoon cells, and I'm trying to represent somewhat both the complexity of the alpha-synuclein aggregation process as well as the fibril seeding pathways. So if we start here with the top cell, what I've represented here is the alpha-synuclein monomer, which is an intrinsically disordered protein in its sort of natural state. And this intrinsically disordered protein self-associates to form oligomers. They can be on pathway, off pathway. They are viewed as a toxic species, as Andy told us. And then we continue somehow to fibril formation, and then there's incorporation. Oh, sorry. Then there's incorporation of these species into the Lewy bodies that are found in the neurons of patients with Parkinson's. And so that's one aspect of the protein aggregation, but another aspect that's really viewed as increasingly important in disease progression is the cell-to-cell spreading of the disease, and that arises when, let's say, fibril fragments, for instance, are released from the cell and taken up by a neighboring cell. So now you have some fibril fragment that's in the neighboring cell, and that cell has endogenous alpha-synuclein and that endogenous monomeric protein. Why does this keep happening? Sorry. That endogenous monomeric protein sees the fibril as a template and can template against the fibril to then have further elongation into a mature fibril, which can again fragment and then move on to other cells. So this process of fibril seeding, where the monomer templates against the fibril, is really important and could be viewed as a therapeutic opportunity. So if you could say, get rid of this arrow here and this interaction, that would really slow down disease progression. And so I don't quite know what's wrong, and I'm very sorry. I'm trying to manage this. And so in today's talk, what I want to focus on is elucidating the molecular underpinnings of alpha-synuclein fibril seeding. And as I was saying, what we're going to be looking at is the monomer interacting with fibrils or fibril seeds and asking what that interaction looks like at the molecular level. And can we figure out what that looks like at the molecular level? And so I want to start by first talking about what fibrils look like, right? Because we're going we're using these fibrils as templates for monomers that are somehow, templating against them to then grow further. And what we know from the PDB is that there are approximately like some 24 distinct alpha-synuclein fibril structures in the PDB. I've just represented some of them here with their PDB numbers, and you see there are wild type fibrils that have been solved. David Eisenberg's lab has been really, really at the forefront of a lot of these structures. So wild type, acetylated protein, and terminally acetylated, and all of the work that I'm going to talk about that we've done is N-terminally acetylated. They've looked at mutant proteins and brain harvested MSA. And what we see is that we have the sort of typical cross beta motif. So these are the fibrils, typical cross beta motif, and protofilaments bundled together with different interfaces. So this is one type of interface, this might be another, and you see these different interfaces here across these pictures. And this gives rise to these multiple polymorphs that everyone talks about in this sort of fibril world. But what I want to point out is that all of these cryo-EM structures really incorporate, or the ordered regions of the cryo-EM structures are really in the sort of central region of the alpha-synuclein sequence. So alpha-synuclein is a 140 amino acid residue protein, goes from 1 to 140. And what we see is that the cryo-EM structures, the ordered regions that are depicted in these cryo-EM structures really are sort of encapsulated by residues roughly 37 to 99. Now, alpha-synuclein is typically divided into 3 sections. When we talk about it, we talk about the N-terminus, which I've depicted in blue, the NAC, which I'm depicting in gray, and the C-terminus in red. The C-terminus goes from about 96 to 140, the NAC, which is really the central core of the fibril, 60 to 96, and the N-terminus 1 to 60. And what we see in these cryo-EM structures with the same color coding is that all of the cryo-EM structures have the gray part. So the NAC is really the core central part of these cryo-EM structures. And you see that many of them pick up a lot of the blue into the N-terminus, but very, very few, if any, have the red, which is about 40 residues long. So you never see red color in this color depiction of cryo-EM. So, but as Andy would, elegantly told us, if we look at the solid state NMR structure, of which there is this one that was solved by Chad Rinstrow with our Andy Newcoop when he was in his laboratory, the solid state NMR ensemble is derived from the Explore NIH computational approaches. And in these ensembles, we visualize the disordered termini. And so it's a different look, but it's a really important different look that I want to highlight, which is when you look at the solid state structure, you see also this ordered region, which is the central core with the gray and the blue. But what you also see are these very floppy disordered termini. And so these disordered termini in the solid state NMR structures arise because, as in solution NMR, there are no restraints. And when you do your MD calculations, these come up as these disordered sort of floppy regions. But what's nice is you can actually see these disordered regions, and we've done that in our laboratory where we've done solid state NMR J-based experiments where you can now, these J-based experiments highlight the mobile disordered regions in the protein. So this is an NF-based carbon-carbon 2D. And what we've done is we've used the assignments that we obtained, that we obtained from a recent paper that was published by Ladinzewski. And so using his assignments, what we see here, and we've blown this up, is that we see the resonances of the C-terminus. We do not see resonances from the N-terminus, which suggests that they are less flexible than those in the C-terminus. And we did a temperature dependence that shows that even at very low temperatures, minus 15 degrees, we still see the intensities that arise from this inept-based experiment, suggesting that it is really hard to freeze out the C terminus, except for the residue that's at the boundary at residue 102. So that's where the cryo-EM structure, ordered core, is sort of based. And then if you go beyond that at lower temperatures, you can freeze that out. So this suggests that this picture of the fibril as having an ordered core, as well as this disordered C terminus, and possibly the cryo-EM structures don't see the N terminus. We don't see it in these inept-based spectra. And so we've sort of depicted them as, semi-mobile. But I think it's really important to think about these fibrils as having a structured core and these disordered regions as well. And so going back to our original question, what are the molecular underpinnings of fibril seeding? We're now going to move, using this picture of the fibril, we're going to move to looking at this through the lens of the alpha-syn monomer. So now we're moving to solution NMR. And what we're going to do is we're going to look at the monomer in equilibrium with the fibril. And so what we do is we set up a situation where we create fibrils that we've labeled with N15. We put them in solution. They're sort of in a suspension, and they actually have an equilibrium where there's an equilibrium between fibril and monomer. And the reason we know this is if we look at an HSQC spectrum in solution, in black, we have the free monomer, which you can't really even see. And in green, we have the monomer that's derived from this equilibrium fibril situation. So this green we'll refer to as the equilibrium monomer so that we don't confuse it with the free monomer that just comes from free alpha-synuclein. So they're fully overlaid, which tells us that we have monomer in solution. Now, these are really well washed, all kinds of controls to make sure that we didn't just have monomer in solution as well. And if we look at the N15R2 experiments, which are a measure of dynamics in solution, as a function of residue, the black is the free monomer, and so the black reflects the profile for the dynamics of the free intrinsically disordered protein, and you see it's really relatively flat. But if we look, we see that in this equilibrium monomer, the green, we see enhanced R2 values in this little region of the N terminus from residues 1 to 12. Suggesting that we have weak transient interactions of the monomer, the extreme N terminus of the monomer with the fibril. We then went ahead and did experiments to further investigate this, and these are called DEST experiments, and they allow you to indirectly probe the monomer interface with the fibril. And the idea behind them is that the NMR bound state, fibril plus monomer, is invisible in solution NMR, but the NMR free state, the monomer is NMR visible, and the monomer retains some memory, in essence, of its interaction with the fibril in a residue-specific manner. And that's what these DEST experiments allow you to do. And if you have an interaction with the fibril, what you see is a DEST profile that's broadened, and I've just shown that here relative to the free monomer. So this is the equilibrium monomer. So we see the DEST profile that's broadened in the N terminus, whereas in the NAC and the C terminus, we see absolutely no broadening. So when we plot all of this broadening, delta theta, as a function of residue, what we see is that the N terminus has this broadening, suggesting that there are weak transient interactions between the extreme N terminus and the monomer, whereas the rest of the monomer is not interacting with the fibril. Okay, so we have a sense of how the monomer is interacting with the fibril, and we know that it's this extreme N terminus here, and in fact, it's the first 12 residues. So if we look at the sequence of alpha-synuclein, again, it's divided into 3, N, NAC, and C, and we look at these first 12 residues. Well, in some ways, there's nothing exceptional about them. The single phenylalanine of the protein is in here. It's got some methionines that can get oxidized, and it's acetylated. And so remember I said that everything we looked at was acetylated. And so what I just would like to point out is that acetylation does impact on the polymorphs of the fibrils. And so in the non-acetylated case, David Eisenberg has shown that you get these rod-type fibrils as well as twister, whereas in N-terminally acetylated protein, Jennifer Lee has solved structures and shows that you get more of this rod-type rather than these more heterogeneous rod and twister polymorphs. And acetylation of the N-terminus also impacts on the monomer. So we showed a number of years ago that when you acetylate the protein, which is in this pink, and you look at secondary structure propensity as a function of residue, what you see is that when you acetylate the protein, you have more residual, you have a propensity to stabilizing transient alpha helix. In fact, in the same first 12 residues that we see the interaction with the fibril relative to non-acetylated alpha-synuclein, which has a pretty flat profile of secondary structure propensity across the residues. This here shows that the C-terminus is quite sort of extended. And we also know that N-terminal acetylation blocks metal binding, in particular copper binding. And it was thought for many years that the site 1, methionine, was a major binding site for copper, but when you acetylate, actually the copper binds the histidine and the C-terminus here. So the extreme N-terminus does seem to play a role in and of course, the extreme N-terminus is the region that binds the lipid membranes and has also been shown to bind chaperones. So this extreme N-terminus really does seem to play a key role in interactions with other proteins, with alpha-synuclein and with the fibril. And so now in this part of the story, we think we understand that the extreme N-terminus is the interface region with the fibril.
Time to wrap up, Jean.
But the question remains, what part of the fibril interacts with the monomer? And so in order to answer that question, we actually looked at the interaction of monomer with fibrils and monomer with off-pathway oligomers. And what we found was that when we made oligomers, we found that we could make them off pathway. They were not good seeders, but Daniel Oateson had also looked at these oligomers and had determined using Sachs that the oligomers have a disordered C-fuzzy terminal coat. And so when we did the same NMR experiments in equilibrium where we looked for interactions of the monomer with the oligomer, we found again that the first 12 residues of the monomer interact with the oligomer. And we were really surprised because we thought, how's that possible? The oligomer is round and diffuse and has a diffuse N-terminus, whereas the fibril, of course, is ordered and elongated. So why is the N-terminus interacting similarly with both the oligomer and the fibril? And we think the reason is that both of them have this outer disordered fuzzy coat. And so what we're proposing is that the monomers interact via the extreme IDP N-terminus here with the intrinsically disordered regions that are flanking the fibril or the oligomer core. And so, this suggests that the weak monomer IDP fibril IDR interactions may serve as the first step in the monomer recruitment by fibrils. So there are obviously further steps that happen so that they can elongate or have secondary nucleation, but this may be the first sort of recruitment step through these intrinsically disordered, transient intrinsically disordered regions. And so lastly, and so I think, part of what we're trying to say is it's important to consider both the core and the flanking regions of the fibril when we think about mechanisms of propagation, seeding, and aggregation. And so I just want to thank my students who've done so much of the work, Snow, who went on to work at Janssen very recently, Cody, a research assistant professor in my lab, John Williams, who just got a really nice job at BMS, John Stober, who did these solid state NMR inept experiments, and my collaborators over the years with whom I've worked on alpha-synuclein, Maral Moradian, Runyan and her lab, Sagar Kare, and Prabhas Mogi, and the NIH.